So today I'll be giving a short lecture on the design of therapeutic bioconjugates, and that's, as you heard, a topic which is very much the focus of my laboratory. And more specifically, how we can use the latest advances in polymer chemistry to tune a new parameter, polymer architecture, and how we can use this as a design parameter for modifying the properties of therapeutic bioconjugates. So within 15 minutes, I have to be selective. So I have a disclaimer, this is a really biased presentation, and I'll be focusing mostly on my own work, and it's not meant to be a comprehensive, lecture on the topic. So the bioconjugates I mostly work with are peptide protein polymer conjugates. So we have a therapeutic peptide or a protein onto which is grown or grafted a synthetic polymer. And the reason why we work with this type of bioconjugates versus others is that approximately 25% of all new drugs approved annually by the US FDA are peptides and proteins. And because of the homogeneity of their chemistry, innovations that you make in bioconjugate chemistry are applicable to many different types of peptides and proteins. And thus, the research we do stands to have an impact in a variety of different diseases. So as we saw in the previous talk, there's a number of challenges when administering bioconjugates, and the route of administration that I'll be discussing today is a systemic administration, so bioconjugates designed to circulate within the bloodstream. In comparison to small molecules, though, proteins have a certain number of different challenges. One is that even though they have a large molecular weight, they're actually compact molecules because they have a compact folded structure. And as such, they can be rapidly eliminated by the kidneys into the urine. Furthermore, many proteins are from non-human origin and as such can elicit immune responses and accumulate and be degraded in the organs of the NPS, such as the liver and the spleen. And furthermore, there are a variety of enzymes in the body which can degrade proteins. So when you administer a protein therapeutic, its concentration immediately increases to a certain value and then rapidly decreases due to a combination of one or more of these phenomena, which means that you have to administer a second dose and a third dose. So obviously, it would be advantageous to go from this situation to a situation where after a single administration, you have a steady concentration within the therapeutic window for a prolonged period of time, avoiding local maxima, which could cause toxicity, and avoiding local minima where you're not having the therapeutic effect. So for roughly 40 years now, the main strategy which has been used to achieve this scenario has been to modify the therapeutic protein with synthetic polymers such as PEG. So pegylation, on one hand, dramatically increases the hydrodynamic radius of the protein because it adopts a random coil, an extended type of structure. And so that slows down renal elimination, or can even prevent it. Furthermore, it acts as a steric barrier for antibodies and enzymes in circulation. So we can achieve protection in this manner. Now, the major dogma of pegylation is that high protection, which is easily achieved by having many copies of PEG or by increasing the molecular weight of PEG, comes at the cost of activity. And much of my work involves modifying therapeutic enzymes, so we have a strong loss of catalytic activity because substrate molecules simply can't filter through. So we rationalized that this was possibly a problem that could be resolved by modifying the architecture of the polymer here on the bioconjugate. So if you pretend this line here is the surface of the protein, when you have linear polymers such as PEG on its surface, because it's a flexible polymer, you have the opportunity of having entanglements between the polymer chains. So this is advantageous to a certain extent because it acts as a steric barrier preventing antibodies from approaching the protein surface, but it also prevents the approach of substrate molecules and the departure of product molecules. So if we could go from this situation to a situation such as these, where we control the aspect ratio of the polymer around the conjugation site on the protein, and if we could avoid entanglements, it may be possible within a certain regime of molecular characteristics to have nanopores between the polymer chains. Pores which are large enough that the substrate molecules can filter in and out if the enzyme catalyzes a reaction involving small molecules, but are sufficiently small that the protein essentially sees just polymer and therefore does not reach the protein's surface. So this looks great on paper. How do we achieve this type of property? We selected a polymer, PEGMA, which is a polymethacrylate with PEG side chain. So it's a comb-shaped polymer. And the reason why we selected this polymer is because previous neutron scattering studies have shown that in solution, depending on the length of the main chain versus the side chain, this polymer can adopt ellipsoidal conformations or cylindrical, an extended flexible cylindrical conformation. This is because of steric concerns and also because of hydrophobicity. When the main chain is very short, it kind of folds upon itself to form a little core, and then the peg chains project out into 3D to minimize their interaction with one another. Whereas when the main chain becomes very long, it does not have room to fold back upon itself, and so it's stretched out, and the side chains project out into 3D, and in the plane perpendicular to the main chain. Moreover, pegma has already been used as a protein repellent coating for solid surfaces, and it's also been grafted to proteins, and it's been shown to extend the circulation lifetime in vivo without any obvious detrimental effects caused by the polymer itself. So I'm not very good in simulation, and it's kind of difficult to predict what kind of molecular characteristics would be necessary to obtain a molecular sieving effect. So we adopted a chemical engineering approach where we just synthesized a large library of protein polymer conjugates. We chose chymotrypsin as a model protein for analysis, mainly because this protein has promiscuous activity, so it can digest substrates of a variety of different sizes. So by analyzing catalytic activity for small molecules versus larger ones, you can actually probe how well these molecules filter through the polymer coating and be digested by the protein. So we installed onto chymotrypsin between 2 and 12 initiators for atom transfer radical polymerization on the amino groups of the protein, of a maximum possible of 14. We then use atom transfer radical polymerization to control the main chain length of the polymer here. And by polymerizing different commercial monomers with different lengths of PEG, between 2 and 21 units, we're able to access a three-dimensional parameter space here. We're able to synthesize over 100 unique protein polymer conjugates differing by one of these parameters here for analysis. So what you see here is it's a three-dimensional representation of this parameter space, where we have the molecular dimensions of the polymers here. So this is the PEG number of units on the side chain. Here we have the molecular weight, the degree of polymerization of the main chain. And in these planes here, we have the degree of grafting, so the number of polymer chains per protein, between 2 and 12 here. Now, the fourth dimension in this graph is color. And color represents the relative permeability of the polymer coatings for small versus large molecules. So when it's red, it means that nothing special is happening, and both small and large molecules are filtering in without any particular difference in terms of size. And this is obvious when we have a low polymer density here, because the catalytic site is really open, so large molecules can come in, come out, small molecules as well. Where it becomes interesting is when we have a really dense coating of polymers here, where we observe this region that's just blue here, where small molecules are able to filter in and out roughly 10 times more easily than larger molecules. At this grafting density, we have roughly one polymer chain for every 5 square nanometers. So the polymers are really close to each other if you consider the molecular dimensions of the monomeric units. And so in order to understand why we kind of had like a hotspot here, which kind of appears with a maximum semi-permeability effect and it disappears thereafter, we then analyzed the conformation of these polymers on the surface of the protein using NMR spectroscopy. And what we observe is that in this region here, where we have the maximum effect, the polymers are adopting these really compact ellipsoidal structures. So the main chain is really collapsed about the conjugation site of the protein, and the PEG side chains are projecting out into 3D, really localized around the conjugation site. So it's really compact in that sense. Presumably that the polymer chains are sufficiently far that we don't have interpenetration between the PEG side chains, and we're creating nanopores here. And this effect dramatically disappears as soon as the main chain becomes long enough that we transition to a cylindrical conformation. Again, presumably, the flexible cylinders are swaying a bit, and we have interpenetration, interdigitation of the PEG side chains between neighboring chains here, which closes this effect. So of course, these are relative values. I'm saying that small molecules filter in more than larger ones. From a more quantitative level, if we take some selected conjugates in this region here, for instance, what we observe is that their catalytic activity compared to the native protein is 100%, okay, despite having 12 large polymer chains on its surface. And furthermore, if we then measure catalytic activity in the presence of a large inhibitor, anti-chymotrypsin, which at 1 molar equivalent completely inactivates the native protein, we have complete insensitivity to this large macromolecule here. So we seem to be achieving protection without compromising catalytic activity. And by analyzing such a large data set of protein polymer conjugates, we really have a good idea of guidelines and things we should target in order to achieve this effect for different proteins. So in vitro analyses, and chymotrypsin is not particularly interesting, aside from the physical chemistry aspects. So then we switched to a therapeutic model, and the model we chose was L-asparaginase, which is an enzyme that catalyzes the hydrolysis of the amino acid asparagine to aspartic acid. So this enzyme is used as a frontline treatment for acute lymphoblastic leukemia, because these leukemic cells are unable to produce asparagine for themselves, and therefore they have to be supplied it by the blood. So when you administer this enzyme in the bloodstream, it depletes asparagine and therefore selectively starves out the leukemic cells. It's a very mild form of chemotherapy. Now, unfortunately, asparaginase is a bacterial origin, and it's known to generate hypersensitivity reactions in roughly 20 to 30% of patients. And in fact, probably for this reason, this is one of the proteins that was first pegylated in the whole history of pegylation. All right. So nonetheless, PEG asparaginase continues to elicit immune responses in roughly 20% of patients. So this is a really antigenic, protein, which catalyzes a reaction involving small molecules. And we rationalized this would be a really interesting model to test how well our strategy works in the adaptive environment of the body. So using the guidelines established for chymotrypsin, we synthesized a small focused library of PEGMA asparaginase conjugates. And our best performer here was one bearing 128 chains of PEGMA 475, which was 3 times less active than the native protein, but 3,000-fold less recognized by anti-asparaginase antibodies in vitro. Okay, so high protection with a low loss of activity. In comparison to a linear PEG control, which had 168 chains of 5 kilodalton PEG, which was slightly more active than our PEGMA asparaginase conjugate, it was only 25-fold less recognized by anti-asparaginase antibodies than the native protein itself. So here we have low protection, but a little bit better activity. So with these conjugates in hand, the first question we asked ourselves was that if these PEGMA polymers are really compact on the surface of the protein, would they extend the circulation lifetime of the protein, of the conjugate in vivo, as would MPEG 5000, which is really as an extended structure with a large impact on hydrodynamic radius. So what we see here by basic pharmacokinetics, pharmacodynamics, is that if you administer the native protein, the linear PEG modified, or the PEGMA modified conjugates, that both conjugates circulate for roughly 2 weeks in about equivalent manner and fully deplete blood asparagine over this entire time. So that appears to not be an issue for this conjugate. The second question we asked ourselves was how well the conjugates were protecting the protein. And for this, we did a sensitization test where we administered 4 large doses of either the protein, the linear PEG modified protein, or the PEGMA modified protein, and we measured antibody titers against the protein part or against the polymer at different times. And what we see is that surprisingly, because in vitro analyses don't always match up, was that we have a good correlation with our in vitro findings, and that in comparison to the native protein, we have a 100-fold reduction of antibody titers for the linear PEG modified protein, and a further decrease of magnitude in the number of antibodies generated towards our conjugate here. Another interesting observation was that we did not observe any anti-PEGMA antibodies, whereas we did observe anti-PEG antibodies. And this is a topic which is increasing interest because of the omnipresence of PEG everywhere, even in cosmetic products. So patients not actually having received pegylated proteins in the past can have antibodies towards PEG nonetheless, just from the products they use every day. So we're continuing this work based on these positive results, and we're currently investigating the influence of polymer sequence as a design parameter for modifying the properties of asparaginase. So if I look back, what are the lessons learned through these studies of polymer architecture? Is that it's quite simple to modify, to control the 3D aspect ratio of architecturally complex polymers using modern polymerization techniques. This can be done on the surface of a protein. In the specific case of enzymes catalyzing reactions with small molecules, dense, compact polymer coatings create a semi-permeable barrier which function in vitro by producing conjugates which have high activity and low antigenicity, as well as in vivo by increasing circulation lifetime and, again, stimulating lower immune responses. So with that, I'd like to conclude by thanking my collaborators and former students, as well as my current research group at INRS, for their contribution to this work and to my research program, as well as funding for our research. Thank you.